Hi, everyone. As part of our comparative genomics series, we're going to start with some of the basic steps that you need to do when you are doing a comparative genomic analysis. One of the first things you'll need to do, or one of the first things we teach, is how to help you use the global search function to create a genome group. So here on the PATRIC homepage, you can see this box here is the global search box. When you're on any page, like let's say I go here onto this page, the global search box occurs here. So it's on any page that you see in PATRIC. So let's first find some things. I've got an example for you. So a list of genomes that you can use to use in the global search box. So let's go to workspaces here and click the down arrow. And then let's go to public workspaces. I'm logged in right now, as you can see here. And you will need to be logged in to do this exercise. So when I'm logged in, it'll look different from what you see when you're logged in, because these are all the workspaces that are shared with me. But we all have this in common. Every user of PATRIC has access to the PATRIC Workshop folder. So let's click on that and open it up. And let's click on the folder that says Protein Family Sorter. Click on that. This is all the Protein Family Sorter is an essential tool in the comparative genomics section. So we're going to be showing that in these series of videos. Today, I'd like you to click on 39 Brucella genomes here. And you notice when I click on anything, any row, be it a table or within a folder in PATRIC, this vertical green bar is populated with a number of icons that indicate possible downstream functions. I can edit the type of file this is, which right now here we can see that it's unspecified. I can move it, I can copy it, I cannot rename, or you cannot rename it because you don't own it. It can be deleted or download. So we want to download this. So let's just click on that. And it looks like it's downloaded. I can find it here. And here's a list of genomes that we're going to use in the global search box. So let's talk a little bit about what these are. These list of numbers are unique identifiers for individual genomes. Now, there are certain things that genomes can be named. For example, Mycobacterium tuberculosis. That's a name for a species, and within that species, there's a very famous strain called Mycobacterium tuberculosis H37RV. Now that name, the name can be broadly shared. In fact, there are currently 4 genomes in PATRIC that are named Mycobacterium tuberculosis H37RV exactly. And there are 14 genomes that have H37RV as part of their name. So across all of those genomes, even though they share the name, this identifier will be unique to them. So that's an important thing to recognize, that this is the closest you can get to any individual genome in PATRIC. So let's copy that number, and let's those numbers. There are 39 Brucella genomes there. And let's go back to the home page, which we can get to by clicking the PATRIC symbol here, just because I like the real estate that is available on the home page for the global search box. So let's paste those numbers into the box, and we're going to ask PATRIC to find those. Let's click here after all data types. Let's click on the down arrow, and it shows you what you could search for. We want to narrow the search. We don't necessarily want antibiotics or taxa or specialty genes. We want to find these genomes. So let's click on that. And then here we have another way to narrow the search. Let's click on that, and we can look for all terms, any terms, all exact terms. Let's click on any exact terms because I want to see these numbers exactly. So click on that. Now we've fine-tuned the search, and we can click on this magnifying glass here that will take us, will launch the search. So very quickly, it gives us the list of genomes. So look, it says there are 39 genomes, and these are the numbers. Let me open that again, and let's look at, so you can see that these numbers here should match these numbers here. One of the things you can do in PATRIC is any genome, you can click, when you're in the table form here, you can click on any of these column heads, and that'll tell you, that'll resort all the data in the table alphabetically or numerically, depending upon that column. So I can do that. You can see I have these number of abortus genomes, a canis genome, bunch of Brucella seti genomes, and Opinata melitensis. So this group spans what we know of as the classical Brucella. So that's the quickest way to use the global search function and with genome identifiers to get a specific list of genomes that you're interested in. So as a practice, there's another, what I would like you to do is go back into the public workspace, and I have a group of genomes called, a text document called Ancestral Brucella. Why don't you try that and see which genomes you get there and see if it meets the number that is in the text file. Keep watching the next instructional video where we'll show you how to create a genome group once you're at this list. Thanks for watching, and thanks for using PATRIC.